as we were saying, we will try to discuss today and actually give some kind of helpful information about the Omics Discovery Index. But we will try to be focused today on searching public Omics data. My name is Perez-Riverol. I'm a project leader at EVI. And these are my contacts: Twitter account and GitHub account. In any lab, this is what we would like to have. This is a really nice picture, a figure from Nature Reviews about all type of omics experiment we can run nowadays that give us insight of the biology of any experiment at different levels. So we can get the genomics data, we can get the modifications at genome level, epigenomics data. We can actually measure also the expression data at the level of genes with the transcriptomics experiment or proteomics. And finally, we can have also some information about the metabolites. And then with all this information, we can infer what's happening in the system biology at the system biology level, phenotype level, and we can infer biological knowledge. However, this is extremely complex because most of our labs nowadays and laboratories, they don't produce all this data. They only produce one specific type of this data. And then the current system biology approach to actually is basically guided by, you get some results, let's say proteomics experiment, from a proteomics experiment, and then some proteins that they change expression. And then you go to, for example, what is called knowledge-based databases like Intact or Reactome to actually make sense out of this data. Which are the interactions that actually are you can infer from your proteomics results. Also, you go to literature to actually get some insights about what is happening actually in the cell or the specific tissue under study. And finally, you will resume all this into a final publication. However, the amount of data is generated nowadays, it makes complex to go from archives to knowledge-based databases. A recent review we have done last year actually said that we have a ratio of almost 100 papers of proteomics and transcriptomics data every month, and the ratio in metabolomics is around 30 data sets per month. Then the current way we store data, it goes through to archive. You know, the data is produced in different labs, is going into archive, and those data is used by these knowledge-based databases to actually produce the biological knowledge. Then there is a lot of interest in the community to add a new component to this, to the current system biology approach, which is the reanalysis of the data. Let's say you have your proteomics experiment, and then you actually do your interaction analysis or pathway analysis, but also you look for some experiments in other omics type that basically can complement your original results. And this is why it's so important to have a way of finding this new data set. I will actually argue that data set it's really different to a publication. I mean, data set, a publication is something static. Let's say you have some results, you basically publish your paper, and this paper will not change on time. Yeah, but a data set, we can say it actually is a dynamic entity. Wherever you can reuse a data set, it will give you different results if you combine with other data sets and so on. The data set value is beyond the original publication. You can have you can do reanalysis, and we have a lot of cases when a reanalysis of the same data has been done, proving that new results come out. Okay, let's say you have a proteomics data set, and then as part of your proteomics data set, you have a set of, for example, you have a study post-translational modification, but in your publication, you're not focused into the post-translational pathways. You basically focus your analysis into the main data. This data can be reanalyzed and integrated with other resources in order to get new claims and new insights into the biological knowledge. And I would say the last thing about the dataset is if one dataset is valuable, but three combined are more valuable. And if you start adding more and more data, then there is a more and more value on it. Okay. What is the outline of this presentation? Is what is the Omics Discovery Index? This is the first thing we want to actually discuss. The second thing is how easy you can search for omics data using OmicsDI. And we will talk a little bit about similar data sets because this is a really important concept. And actually, at the end, we will talk about new features in OmicsDI like data reuse and citation. Okay? How someone right now that has some genomics or transcriptomic data can find, for example, the proteomic data set, or the way around. The way to search for data right now is to go into a specific databases that or archives that store those data sets, or you can go to what is called consortium web page like Metabolome Exchange, Protein Exchange that aggregate a set of a subset of databases. You can also, a lot of people try to go through the publication path. Let's say you go to PubMed, find some really important publication for you, and then try to find out if the data set associated is public or not. But a lot of people will end up in Google by basically typing some keywords and trying to find out if there is any data set around that. This is a problem that has been solved for many years, but for example, in the publication side by European C and PubMed, that where researchers go there to find a centralized point where all the biological publications can be found. But there is no place like that for data sets. And you can imagine as a user, okay, because that the problem is that in the data set concept, there is not that much of resources. We did actually, in fact, it's not true. I mean, in 2015, we did a review about only protein expression resources. And you can find here, we have more than 13 resources that have been created in the last 3 or 4 years. And some of them are aggregated within consortiums like TRI, Massive, or Peptide Atlas, by others no. And there is a lot of valuable data that can be found, for example, in MOPED, GPNDB, COPA-KB, PAXDB, and other databases. What to do then? I mean, in the last 2 years, we have been implementing a central point where we can actually aggregate data coming from multiple resources in a centralized way that help the user to find, query, and link the data to the original providers. And this is actually the Omics Discovery Index is OmicsDI. These centralized points actually aggregate right now more than 15 databases in 4 different continents. We have more than 90,000 data sets coming from, for example, proteomics. We have the major partners from Protein Exchange like TRI, Peptide Atlas, Massive, GPNDB, but also we have a lot of transcriptomic data like ARRA-Express, Geo, Expression Atlas, and also metabolomics data and genomics data. How you can search in OmicsDI? I mean, the first the more easy way to search for data is by looking into the resource and then do some queries, as we do, for example, when you were looking for other things in our life, let's say in eBay or Amazon. You can basically use the search box here to type your query. And there is a lot of help provided by OmicsDI that enable you to filter out and actually refine your query. Let's say you can search here for a couple of keywords, like, for example, proteomics, or you want to study for example, cancer, and so on. And then here, in the left side of the web page, you can filter out or refine your query, for example, by a specific organisms, or a specific tissue, or a specific disease, and so on and so forth. We have, for example, the major features that we provide right now are, for example, organisms, repository, tissue, disease, type of omics data that can be found here, like proteomics, metabolomics, and so on. But also, you can filter out about specific molecules. And I will only say right now in this slide that you can search by a specific molecule, let's say one specific protein or one specific gene, and we will let you know how much you can do with that. Also, you can actually sort your results by accession. Accession is the data set accession, but also the date that this data set has been published in the original resource, because OmicsDI, and this is a really important concept, do not store the data. OmicsDI only aggregate the data coming from different places and try to provide a way of searching into the data. Then the publication sort can be used to sort it out your results by the publication date in the original provider. And finally, the relevance, which is actually how close is your query to your specific results. Okay. But we provide more. I mean, because it's a search system, you can actually, we actually help the user to do autocomplete in the search box, and then when the user is typing, then OmicsDI will provide a set of keywords that will help the user to construct their sentence, taking into account the data that we have. And there is another really important feature, is that OmicsDI behind the scene has a lot of pipelines that help the user to find out datasets that can be part of one specific or more broadly study, like a multi-omics study. And this is why we have this icon for some of the dataset when a dataset has been named as a multi-omics dataset. And what is that? Basically, imagine you have your study, and then you have some proteomics data as an output and metabolomics data. All these proteomics data will go into, for example, PRI database, which is in the EBI. And let's say the metabolomics data will go into Metabolome Express, which is in US, okay? Or Metabolome Workbench, sorry, which is in US. And then in OmicsDI, and then both of those data sets are related with one specific publication, yeah? And then OmicsDI has a pipeline behind the scene that link both data sets. And then when a data set is actually named as a multi-omics data set, it's because there is some other data coming for a different type of omics in another repository that you can be used, okay? We will explain with a really nice example after this slide. But we also provide advanced search, and this is one really important feature in OmicsDI. You can use the advanced search to actually improve your query capabilities, but also, for example, to search by, I would like to search by these uniprot proteins and search all the proteomics data set with this, where these two proteins has been identified at the same time, okay? And then you can do this with gene. You can actually search for all the genes that change the expression in all the data set that OmicsDI contain, and it will actually boost all the data set where these two genes change the expression. I need to say right now that actually the search capability for biological entities end up by saying this molecule, this specific molecule has been identified or change the expression within this particular data set, and that's it. Not the actual sign of this change, if it's overexpressed or downregulated. It's only basically saying, pointing the user to, okay, these two particular molecules, or 3 or 4, whatever you want to query, are changing the expression or they have been identified. Okay, then we have more capabilities. I encourage you to play with the resource. But OmicsDI do not stop there. We actually provide a new concept, which is actually similar data set. Let's say you search for some data, and then you find one specific data set is interesting for you. We actually provide similarity metrics around each specific data set to highlight other data set within OmicsDI concept that can be close or interesting to you. In the same way, for example, Amazon, eBay, or other resources basically suggest different products to you when you're actually taking a look to one specific, the product of interest. And we do this at a different level. The first level is at the level of the metadata. Basically, by comparing all the metadata across OmicsDI, we can actually highlight all datasets that are close or similar to your dataset. We also do this by direct publication, and this is what we were saying about multi-omics. Let's say two specific studies in different databases are related with a specific publication, then we link those datasets because we know that these datasets are close because they are related by the same publication. And finally, by the molecules. And this is actually a really important concept we have been trying to work for more than a year, proving and getting excellent results. I mean, for each specific data set, we collect all the molecules that are interested for this data set. Let's say if it's a proteomics data set, will be protein identifiers. If it's a transcriptomic data set, would be genes identifier. If it's a metabolomic data set, the metabolites identifier. And then we cross-link those lists of identifiers and detect what are the common similarities between these molecules. I mean, which are the intersection within those lists to highlight which are the data sets that are more close, taking into account only the biological entities. And these are, for example, some examples. Then when you are in OmicsDI and then you check this specific data set, for example, or other data set, you will have in the right a box about what are the data sets that are similar to your data set by the metadata. And these are the related data set by metadata, okay? For example, this is a data set in ArrayExpress, and here highlighted all the geo data sets that are in ArrayExpress also that are close to your data set by taking into account only the metadata. But you can actually, and for example, we do this also with as I said, with the publication. This is actually a data set that is in Pride and also but the related data set in the same publication is in ArrayExpress. And then you have here, this is the data set in Pride, the one that we are actually having a look, and then annotated with the multi-omics, because I explained that when two data sets coming from different omics types are linked by the publication, they are annotated as multi-omics. And then the other omics data sets in OmicsDI are that this actually a different type of omics is actually annotated in is annotated here, and the link can be found here. If the user click here, will go directly into the ArrayExpress dataset. Okay, and the final thing is about the biological entities. Let's say this is another dataset, the Pride dataset, and these are all the datasets that share with a high score higher than 0.5 molecules. The same proteins are changing the expression, or in this case, they are identified in those experiments. And then if you move here, the threshold, you will see more the ones that are more closer and closer and closer to your data set. This is actually really important because these three features are completely orthogonal. I mean, it's not the same what you put in the metadata, because remember that in the metadata you can be talking about, for example, the instrument, about the tissue, and so on. But actually, the biological entities measure which are the molecules that change the expression or are identified in a specific data set or biological system. And with that, I will end up what you can do actually searching into OmicsDI. I encourage you to play with the resource and to search, and also came back to us with comments about how to use the resource and which type of feature do you want that the developer team add to the system. But let's start with something we have been working for a couple of months about data reuse as citation. There is a lot of discussion about how to give value to the data. and we would like to contribute with that because we have all the omics data. We want to work, we have been working in 3 different directions, because wherever you find a data set, you would like to see, okay, if I download this data set or take my time to download this data set, I would like that this data set actually not only match with my query, but also have some value, yeah? And this value needs to be compute in the resource. I mean, it's something that needs to be provided to the user. We are actually computing this or calculating these values in 3 different ways, in 2 different ways, tracing the reuse of the data by the reanalysis and the citations. And the other thing is tracing the number of views and access to each data set. And finally, also, we'll talk about how much you can do with OmicsDI to get credit around the data that you publish. The first thing is that for every data set in OmicsDI, we actually trace all the data set that reuse the original data. For example, this is the draft of the human proteome, and this is actually stored in PRI database, and this has been reanalyzed by another proteomics database called Peptide Atlas in all these experiments. Then when you arrive to this data set, you will see the number of reanalysis that this original data set has. And this give you actually, this will give you an impact of how many times one data set has been reanalyzed by other users, or has been used by other users rather than the original authors. The second thing is that we are now providing a way of citing, you know, because it's not only if we want to highlight the value of the data, not only the value of the publication, because when you cite a publication, you can be talking about, I don't know, one specific method in the publication or one specific picture, or you can be against actually the publication or citing because it's an important method to use. But when you cite, but we want to actually have a direct link between the data set reuse, and by that, I think it should be implemented by citing the original data set. Then for every data set, you have a button that says cite, and then user can go here and copy the data set citation and add them to the original, to their publication when they reuse this data set. The second thing we are doing right now is actually tracing how many times this data set has been seen or has been checked with from the user of OmicsDI. This will enable in the future, for example, to rank the original, the search by the number of views and also citations of the specific data sets. Also, users can comment. I mean, at the end of each data set, user can go there, see the publication. We put here the information around the publication, and then user can log in and comment in each data set. And but we want to, we have been working also in a way of getting credit around your public data. A lot of these data sets are disseminated, and it's difficult to trace them. It's similar to what you have with Google Scholar for your publications, but we would like to have it also for the public data. And this will enable actually to give an impact about how much data do you publish and which type of study do you normally do, and actually enable also to user, and this has been requested by many users, to have a way of listing all the data sets that have been making public by individual labs. We want to boost that our data get not only public through the publication, but also public the raw file and the original data for reproducibility purpose. When you go to OmicsDI, you can go and see a login system. Then right now support ORCID, it will support soon Elixir, and it's supporting right now also Twitter, GitHub, and so on. When you log into the system, you will see a profile like this, a profile page that you can update with some information. It doesn't need to be, it's not like it's not like ResearchGate when you need to specify a lot of information. So it's only that we need some specific information that can be pulled even from ORCID. When you have your homepage, your affiliation, and some description about you. And there is a small box here that you can actually set, I want my profile public, like you have, for example, in Google Scholar. Then you can start by adding your data set. And this is the way to go is actually you go to your search box in OmicsDI and type your name and last name. Yeah, and it will appear all the data sets that are related with this keyword in some way, which is actually your name or last name. I need to say that you can use combination of them depending of the how the data set have been annotated and the publication, and then probably you need to use only the last name and so on. When you click a data set that you want to add, the first thing is that you need to click the data set to check actually that this is your data set. Okay. And finally, at the bottom, you have a small link called Claim Dataset, when you can actually click there and add this specific dataset to your profile. The dataset will be automatically added to your profile, and it will be listed with all your dataset. Also, we have added a contact information when you have all the information about your affiliation, your ORCID ID, and also your social connections, because if you log in with ORCID and then you log in with Elixir, we actually managed to merge both accounts. Finally, if you want your account public, I would say you have a link like this here that you can reuse to share with your friends, or you can put in your lab page when you can actually list all your data set for your laboratory. I mean, the important and strong thing here is that you can add all the data sets for your lab or for your personal profile. It can be added into CVs or into web page labs and so on and so forth. Some statistics. I mean, we have been tracing the last couple of months statistics. We have been very lucky and very well accessed. We have in total almost 12,000 users, new users around 10,000, 2,000 are returning and reusing the resource for their analysis and looking for new data and so on and so forth. We will trace this, and then probably at some point we will make public in our web page all the statistics about how the resource is used. I would like to finish with a couple of conclusions. OmicsDI provides a unique resource for omics dataset searching. It's not only for genomics, but also proteomics, metabolomics, and transcriptomic data. With metadata and biological entities and also publication, it enabled to find similar data set across different public repositories. And with we can now trace the reuse and citations using the OmicsDI service. And I will encourage you to finally subscribe to OmicsDI and add your public profile, and also start adding your data set and share with your coworkers and collaborators. Thanks for your time and questions.